Welcome to my presentation. I am Shubho Ghosh, a master's student in the Department of Environment and Energy Engineering, Cheonam National University, South Korea. My selected article is Assessment of Microbial Fuel Cell Configurations and Power Densities. The article is published Journal of the American Chemical Society in 2015 by a group of scientists. Let we see the contents in this study. At the beginning, we will see general information about microbial fuel technologies and microbial fuel cells, then different types of microbial fuel technologies and their application. Next, we'll discuss about the application of different microbial fuel cell based on power. After that, we try to explain power production per cathode area based on the volume of the microbial fuel cells. After that, we will discuss about modular design of microbial fuel cells. Finally, we will summarize this study in discussion section. First of all, introduction. Microbial fuel cells, MFCs, are devices that use bacteria as the catalysts to oxidize organic and inorganic matter and generate current. Sustaining nature of the microorganisms and versatility in the chemicals made this technology more advantageous. High cost of membranes will likely limit applications that might require a membrane. There are still concerns regarding whether larger scale systems can provide power densities comparable to those produced on laboratory size scale for microbial fuel cells, which do not require a membrane. The careful consideration of electrode spacing and packing per unit volume of the reactor is required while scaling up systems. Next, different microbial electrochemical technologies. Among different fuel cells listed in the Table 1, microbial fuel cells and microbial electrolysis cells lacking membranes. 2 or more membranes are used in other types of microbial electrochemical technologies for different functions. For power production based on different applications, from Figure 1, we can see different sizes of microbial fuel cell based on power production. Photographs or schematics of different types of microbial fuel cells. Figure A, microbial fuel cells. Its anode volume is 25 microliter. Figure B represent a microbial fuel cells with air cathode and reference electrodes used in many different laboratory studies, shown here with a brush anode. While Figure C is a sediment microbial fuel cell and Figure D showed a 3-brush electrode microbial fuel cell designed for continuous flow. Finally, Figure E and F represent larger scale microbial fuel cell and baffled microbial fuel cells respectively. Microbial fuel cells have been constructed at a variety of scales, from microliters to hundreds of liters. Next is power production per cathode area based on the volume of the microbial fuel cells. In the Figure 2, we can see power production per cathode area based on the volume of the microbial fuel cells. Power densities normalized to the cathode projected area. Microbial fuel cells that used defined substrates produced more power at smaller scales rather than at large scales. Also, power densities depends on reactor design and operating conditions. In Figure 3 is power production per cathode area based on the volumetric power density. Microbial fuel cells using defined substrates had clearly outstripped. Microbial fuel cells, wastewater, results on the basis of volumetric density. Microbial fuel cells' power density have no clear trends in terms of volumetric power production. Next, power production per cathode area based on the cathode-specific surface area as a function of microbial fuel cells' volume for all data. In the Figure 4, we observed power production per cathode area based on the cathodic-specific surface area as a function of microbial fuel cells. Electrode spacing is a critical factor in power production when scaling up microbial fuel cells. When both electrodes are put in a closed configuration, then produced more power density. On the hand, the packing density shows a general decreasing trend with a larger reactor size. Next, power production per cathode area based on the cathode-specific surface area as a function of microbial fuel cells volume for all data. In the figure 4, we observed power production per cathode area based on the cathodic-specific surface area as a function of microbial fuel cells. Electrode spacing is a critical factor in power production when scaling up microbial fuel cells. When both electrodes are put in a closed configuration, then produced more power density. On the hand, the packing density shows a general decreasing trend with a larger reactor size. Production per cathode area based on microbial fuel cells and microbial fuel cells using wastewater, in the figure 5, represent the power production per cathode area based on microbial fuel cells using wastewater. A general decreasing trend with a larger reactor size is less clear for just microbial fuel cells using single substrates and wastewater. Next is modular designs of microbial electrochemical technologies. Commercialization of microbial fuel cells will require mass manufacturing of the materials in a modular format. The success of the design will depend on many factors. The cathode-specific surface area must therefore be defined for each module design. The mass production of the components in a modular manner will be necessary for the commercialization of microbial fuel cells. The bigger reactor won't operate as effectively as the smaller one. The lack of sufficient cathode-specific surface area will result in poor volumetric power densities for bigger reactor sizes if cathode-specific surface areas are not maintained. Finally, discussion section. In the near term, the most promising microbial electrochemical technologies, MET, applications will likely be microbial fuel cells, MFC, for wastewater treatment, as these systems lack expensive membranes and can accomplish removal of organic matter without aeration, and sediment microbial fuel cell as remote power sources. Successful scale-up will require maintaining high cathode-specific surface areas to maximize volumetric power production and organic matter utilization rates. With successful commercialization of microbial fuel cells, and in the future the availability of inexpensive membranes, it may be possible to further develop other microbial electrochemical technologies. The ability to manufacture reactor cathodes is currently the biggest barrier to the commercial production of MFCs. High cathode-specific surface areas must be maintained for scale-up to be successful. Other renewable energy sources may benefit from the development of low-cost membranes. Cathode will be needed for reactors in the hundreds to thousands of square meters. MFC commercialization presents an economic opportunity as well as a production difficulty. Thank you for watching. Please subscribe the channel and enjoy. My questions related to this article is, Question 1. What are the advantages of microbial electrochemical technologies? Question 2. How microbial fuel cell is different from other microbial electrochemical technologies? And, Question 3, what are the key factors in power production?